the effect was quite astonishing, and also a colleague all over the world, when they saw the results, they were quite surprised.
Nicola Vannini is a researcher in the Department of Oncology at the University of Lausanne in Switzerland. In this episode, we continue our exploration of the gut metabolite Urolithin A, and evidence that this natural compound plays a key role in the health of our immune system and our ability to fight infections. In this conversation, we focus on a particular type of stem cells that are important for the health of our blood and immune systems. They're called hematopoietic stem cells, and as we age, we know their ability to do their job declines. The question, and subject of the new research we're about to discuss, is can that decline be slowed down or even reversed through exposure to urolithin A?
We did two sets of experiments, one ex vivo, so essentially where we took hematopoietic stem cell from old mice and we culture in a dish in the presence of urolithin A, and another set of experiment where we fed the mice and then we checked the immune functionality. So in the first set of experiment, we took hematopoietic stem cell from young old mice, and the one from old mice, they were treated with urolithin A. Afterwards, they were transplanted in recipient mice, and the results were quite astonishing because all the hematopoietic stem cell treated with urolithin A, they perform as well as the young hematopoietic stem cell. So it had the same capacity to reconstitute the blood and immune system of a lethally irradiated mouse. In the second set of experiment, we supplemented urolithin A in the food, and we tested the capacity of these mice to respond to a virus. So essentially, we infected the mice after a feeding period of 3-4 months, and we measured the quantity of the virus in the mouse. And the mice that were fed with urolithin A, they were much better in clearing the virus. So they had much better response to the virus. Not as well as the young mice, but they performed much better than old mice that didn't receive urolithin A.
And this was a synthetic form of urolithin A that you are using. And just for clarity, urolithin A, it comes in one form, whether we're talking about mice or humans, it is the same. It is produced in our gut, but we're talking about the same compound.
Yeah, exactly. It's the same compounds, and it was chemically synthesized. Essentially, we use a compound that is chemically synthesized, and we provide both or in the dish when we culture the hematopoietic stem cell for a transplantation, or we provide it in the food of mice. But the molecule is the same, the one that you have in mouse and the one that you have in human.
And you described the results as astonishing. To what extent were you astonished, and what are do you think, the implications of this?
So the results were quite surprising because it's for me it's several years that I'm working on different molecules that are known to modulate the hematopoietic immune function, but it's the first time for me that I found a molecule that has such strong effect. And obviously the impact is quite wide, because you can imagine that improving the immune function of older adult has a really big impact, because we know that elderly population are more susceptible to infection and cancer development, and we can imagine that by boosting their immune system with this with urolithin A, we can prevent infection, but also we can delay the onset of cancer.
Which is really quite striking, isn't it? Now, what is the next stage? There have been some human studies, presumably more clinical trials involving people are needed.
Yeah, so we are now trying to set up our own trial with a specific category of patients that have similar feature of immunosenescence that you can find in elderly population. And this category of patients are cancer-free patients that receive chemotherapy, because it's known that chemotherapy is inducing premature aging of the immune system. So now this effect is permanent. So people that have received chemotherapy, even they are cancer-free, they are cured, but the damage of immune system will persist. So the idea is to see whether urolithin A can revert these immunosenescent features also in this category of patients. And the other like direct application that I can see for urolithin A is also in the context of elderly vaccination, because it's known that elderly, they respond much less to vaccine immunization. So we can think to supplement urolithin A to this category of patients before receiving vaccine to have a more long-lasting response.
So in addition to, and I frame it like this, reversing aging, or at least reducing the biological age of an individual animal, the applications, as you've described, are quite wide in terms of not only preventing disease, but treating disease as well.
Yes, so I can see both the things, because you can imagine that if you boost the immune system, you can, in an elderly population, you can prevent infection, the onset of cancer development, and but at the same time, being a molecule that restore the mitochondrial function, there are many diseases that are that carry mitochondrial mutation. So we can also think to apply this kind of supplementation in the category of people that have this mutation in mitochondrial machinery. So in a way to restore the metabolic fitness of the cell.
Just one final thought. What kind of response have you had from the scientific community, and especially researchers involved in this particular field outside of your own laboratory? What has the response been to this study?
So they actually, the effect was quite, as I say, was quite astonishing, and also a colleague all over the world, when they saw the results, they were quite surprised, because it's for me and also for many of my colleagues, it was the first time that they see a molecule that was able, especially in the context of transplantation, to restore completely the blood reconstitution capability. And these results, they were presented also at the ASH meeting at the American Association of Hematology last year, and I had a really nice feedback from many colleagues. So I think the story was quite well received from the community.